Hello, and welcome to the Cancer Research Institute Cancer Immunotherapy in You patient webinar series. Today is Tuesday, June 26, 2018, and the title of today's webinar is Revving Up T Cells to Fight Cancer, Cellular Immunotherapies. Before we begin, I'd like to thank our generous sponsors who have made this webinar series possible. Bristol Myers Squibb, with additional support from Regeneron, Sanofi Genzyme, and Adaptimmune. My name is Arthur Brodsky, and I'm the science writer at the Cancer Research Institute, a nonprofit organization established 65 years ago with the mission to save more lives by funding research that aims to harness the immune system's power to conquer all cancers. This work has contributed to the development of life-saving immunotherapies for a variety of cancer types. We present this webinar series to patients and caregivers to help them understand what immunotherapy is and how it differs from other treatments, to provide information on the latest developments in research and treatment, and to connect patients to immunotherapy clinical trials. Now, it is my pleasure to introduce today's expert speaker. Dr. Philip Greenberg is the head of immunology at the Fred Hutchinson Cancer Research Center and was a founding member of the University of Washington Department of Immunology, where he remains a professor in medicine and immunotherapy. In the late 1990s, Dr. Greenberg was part of the team that first revealed that patient-derived T cells could be used against disease. After first demonstrating this approach's effectiveness in patients infected by the cytomegalovirus, his team later showed that it could also benefit melanoma patients and patients with acute myeloid leukemia. Since then, through both laboratory research and clinical trials, he has advanced our ability to re-engineer T cells and make them even better at fighting cancer. In recognition of his achievements, Dr. Greenberg received the 2011 William B. Coley Award and has been elected to the American Society of for Clinical Investigation, the Association of American Physicians, and the American Association for the Advancement of Science. He is also currently a member researcher affiliated with the Parker Institute for Cancer Immunotherapy, as well as a member of the Cancer Research Institute's Scientific Advisory Council. Dr. Greenberg, it is an honor to have you with us here today.
Thanks very much. I thought we would start out with a general sense of what do we mean by cellular therapy, what are we trying to do, and then get into some of the more specifics about how cellular immunotherapies actually work. So the first issue is that cellular immunotherapy, and the ones that we're really talking about, are all focused on manipulating T cells, one of the cells of the immune system. So we need to actually ask, what are they and where do they come from? And I'm trying, can I advance this? Yeah. So the issue is that T cells are just one of the cells of our blood system. They come from a single progenitor cell that's in the bone marrow. And that single stem cell is able to make all of the white cells in our body, in fact, all of the red cells in our body, as well as all of the platelets in our body. They all are essentially blood-formed cells. But there's this particular subset, which are the T cells. And those cells, as they evolve from the stem cells in the bone marrow, need to actually mature to become functional. And they do that by migrating from the bone marrow through the blood into the thymus, which is an organ that's located here up in our neck and is found quite abundantly when we're young and tends to atrophy, unfortunately, some as we get older. But it's very efficient at actually educating these progenitor cells, progenitor T cells, into becoming mature T cells. And they develop in the thymus, and in the thymus, what the special function that's performed is cells that are potentially dangerous and would recognize cells in our body and cause autoimmunity are deleted, and cells that will recognize something that's different from what's normal in our body are allowed to leave the thymus and populate the blood and the lymph nodes and essentially provide the basis for us to have an immune response if we encounter a target that's abnormal from our normal cells or different from our normal cells. So T cells are not surprisingly, it's not that simple. There are multiple types of T cells. We're going to focus on a particular type, but there are what we call killer T cells and helper T cells, which essentially help those T cells, help also B cells, which make our antibodies. There are more primitive T cells that perform what we call innate immunity, which more generally recognize things, but don't have the fine specificity of killer T cells and helper T cells. And these are the gamma delta T cells and the natural killer T cells. And we're not really going to talk about that. We're going to focus on this particular population, killer T cells, because this is really where cellular therapies are currently focused. Although increasingly we're going to be adding to that the use of helper T cells or engineering, as we'll talk about, killer T cells, to be able to form the functions that helper T cells normally provide them and be able to perform them themselves, so we don't need actually two cell type. But we'll get to that a bit later. So what are killer T cells? So these are what we commonly call the foot soldiers of the immune system. And they are the foot soldiers because they're the cells that go in and recognize and then kill infected or cancerous cells. So they're essentially the cells that are made to do the end job, the effector cells, as opposed, as I alluded to, the helper cells, which essentially help these cells expand and become functional. And they recognize foreign cells or infected cells or cancerous cells because one of the things that they develop when they're maturing in the thymus gland is a T cell receptor. It's the receptor on T cells that essentially enables to have precision and to target a specific antigen. And so every T cell in our body has its own T cell receptor. So they recognize different things. And we're talking now about tens of billions of cells that all recognize something different. And then some of these cells, as they proliferate, they form daughter cells that retain that same T cell receptor so that we have more than a single cell with the same receptor, but we have this huge diversity. So it's the reason why, for example, our immune system can recognize flu virus and also recognize cytomegalovirus or HIV or smallpox and be able to recognize all these differently because different T cells with different receptors have unique specificities for those particular pathogens. Of course, that's what we want these cells to do in the context of recognizing tumors. So how does this immune response start? And I think this becomes an important issue to try to understand how we get to having a functional immune response. So normally for a T cell to, after it leaves the thymus, it's what we call naive. It's not responded to any antigen yet. It just leaves the thymus with the capacity to function and with the receptor for specificity. So it needs actually to see something that tells it that it's recognized something and needs to initiate a response. And that initiation is done by antigen presenting cells or dendritic cells that are what we call professional cells that are made to present antigens to cells of the immune system and activate them and drive them to proliferate and differentiate and then go out and recognize their target. And so these dendritic cells essentially, whoops, I'm sorry, I lost that for a second. These dendritic cells take up dying or dead cancer cells. So cancer cells, as they're dividing and expanding, some of the cells outgrow their blood supply or outgrow the nutrient supply or just get crowded out in a large tumor and they die. They can also die because of an immune response or because of chemotherapy. But regardless of the mechanism, these antigen-presenting cells have receptors on them that recognize dying and dead cells and take up those cells by initially binding them and then engulfing them and taking them inside the cell. Now, once they're inside the cell, the antigen-presenting cells now degrade those the cell that they've taken up or the particles that they've taken up, and they break them down into very individual components, which we call antigen processing. So they essentially take very large, complex cells and molecules, and break it down into very small molecules that they can then use for a T cell to recognize. And it does that by putting that little piece of the target into a molecule called major histocompatibility complex class 1 molecules. There's also class 2 molecules in which they put proteins into those molecules, and that's for helper T cells to recognize. But killer T cells recognize these class 1 molecules with these very small proteins, typically 9 to 11 amino acids that are put onto the cell surface. Now, there are many, many class 1 molecules on the surface of a dendritic cell, and they contain thousands, potentially millions of different peptides. And so a T cell needs to actually scan this whole dendritic cell to see whether its receptor actually recognizes that. And so what happens is a T cell comes along with its receptor, and it interacts with this antigen presenting cell. And if it doesn't find anything that the receptor binds to, it just leaves. But if by chance it actually finds a molecule that actually contains this protein that its receptor is specific for, it binds to it, and that binding results in a series of events that are initiated now by the antigen presenting cell, the dendritic cell, which activates that T cell, so that this activated T cell now is no longer what we call naive, but is now been triggered to become activated and become a killer T cell. So it now acquires effector functions. It also divides and produces several more of itself so that there's more than a single killer T cell to go out. And after it's done this, it then leaves the lymph node where all of this has been occurring. And it goes out and patrols and looks for potential targets. And so it's now activated and capable now of mediating effector functions such as killing, but it needs, again, to be triggered. So it leaves the milieu of the antigen presenting cell, circulates through the body, and scans every cell it interacts with, and again looks to see whether it will ever find another cell with a MHC molecule on it that expresses that distinctly same peptide antigen. And if it does, it binds to it. But now, because it's been activated and has effector functions, that interaction has a very different outcome. So that now triggers the activated killer T cell to release its kilogranules and then to kill the cancer cell. Now, that is the way we want a natural immune response to work, that it would actually recognize efficiently a tumor cell, it would target it, it would then eliminate it, that it would be able to expand and now go out, now leave, recirculate again, and keep looking for more cancer cells so that it can not just eliminate a single cell, but become able to eliminate more. In fact, it's one of the reasons many people have nicknamed serial killer T cells serial killer cells, because they are serial killers. They don't kill just one target. They're able to leave and go recognize other targets. And that's one of the great advantages of the immune system, is it's essentially a living drug. It not just kills, but it also expands and looks for more targets, and then looks for more targets to try to continue to make sure that it's eliminated everything that it can bind to. Now, the problem is that if this worked really well, we wouldn't have cancers. And so there's a reason why this isn't so good for cancers. And one of the reasons is because the immune system developed not to recognize tumor cells, which are essentially normal cells in our body that have gone awry. They've acquired some changes that make them, unfortunately, behave in a malignant fashion, but they still are derived from a normal cell. So they're not very different from our normal cells. And as we talked about, when T cells are developing in the thymus gland, what they're learning to do is to recognize things that are foreign, not recognize things that are self, that look like a normal cell, because that would give us autoimmune diseases. And so the problem is that the immune system is very efficient at recognizing cells that have been infected with a virus or a bacteria, because that contains a completely different set of proteins that are brought in by the virus and the bacteria. So these have different genomes, they have different genes, they express different proteins. They're completely foreign. And so T cells are really good at doing that. But the problem is that those kinds of antigens are just not expressed in abundance on cancer cells. So we have this problem that the tumors, when the antigens get taken up by dendritic cells and presented, they are not very different. And so there's not a lot of T cells that can recognize it. So part of it is that there's not enough antigens provided by these dead tumor cells to effectively trigger a response. In addition, tumor cells bring with it, when they're taken up by a dendritic cell, bring along molecules that interfere with the ability of these antigen-presenting dendritic cells to efficiently activate T cells. So there's deficient antigen presentation. So unlike when viruses or bacteria cause inflammation and make a dendritic cell really effective at triggering and activating a T cell, tumors interfere with that. And so we get limited antigen presentation and poor T cell activation. So again, the natural response to the cancer is not terribly efficient. And then finally, these T cells then, even if they've essentially gotten effectively activated and triggered, and they leave and they go patrol and look for a tumor, tumors have developed strategies to interfere with the ability of a T cell to either recognize it or to kill it or to expand or to become a serial killer after it's actually recognized a single tumor cell. So tumors have evolved a lot of evasion strategies that interfere with our ability to have an effective natural immune response. Now, it doesn't mean the immune response can't work, but it does mean that we probably are going to need ways to enhance the way the immune system is recognizing tumors and killing it. And indeed, that's where the whole field of immunotherapy has stepped in here and said, okay, there is an immune response to be had, but how can we make that immune response stronger? And that has been really the revolution of immunotherapy, that in the last decade, we've really learned how to step up that modulation of a weak or poorly or non-existent immune response and make it much more potent and capable of recognizing tumors both effectively and safely. And that's really been part of the huge advances in the last decade. So we're going to focus on a particular approach, which is adoptive T cell therapy, and which is the area that my laboratory has really focused on. So in this context, what happens is we take cells out of the body of the patient, and as you can see, it's a T cell that has a receptor. We now stimulate it outside the body in a setting where now we can efficiently provide good activation. We can stimulate the dendritic cell to be a very good stimulus, and we can provide the right antigen on that dendritic cell to stimulate the T cell. So by making a really good dendritic cell with the right antigen, we can take a lot of T cells out of the body and stimulate them, and the ones that have that specific receptor will get activated, and they then will expand, because that's the strategy, that's the way antigen presenting cells trigger a T cell. So now, instead of having a single cell, we can expand it to have a larger number. We also can add growth factors that stimulate the expansion of those T cells so that they become much larger in number. And then we reinfuse that back into the patient. Now, this is something that works extraordinarily well in settings in which, as you might imagine, there are T cells that effectively can recognize the tumor. So this has been very good. We put these cells back in, and they seek out and destroy tumor cells. Now, this works, again, for tumors in which there are lots of antigens that can be efficiently recognized. So, for example, this works very well in settings like melanoma, and I think we're going to show an example of that. So where we take cells out of patients with melanoma, You can expand them and put them back in, and they go out and seek the melanoma and kill it. And there have been dramatic responses and cures by such cellular therapy against melanoma that our lab and many other labs now have done in melanoma patients. And the reason that works so well is that melanoma, as you know, is a skin cancer, and it arises largely because of UV irradiation from the sun that induces mutations in the melanoma that makes it different from a normal melanocyte, a normal progenitor cell that ultimately leads to melanoma. And some of those differences ultimately are what causes it to become a cancer. But it acquires all of these mutations which encode for slightly different proteins. So melanomas typically have more than 1,000 different novel proteins it's expressing, much like essentially a virus or a bacteria. And so it's possible to find cells that recognize melanoma and to put them back in and treat patients. And labs and groups such as, for example, the Rosenberg lab at the NCI have developed these protocols called TIL therapy, where they take out a biopsy of the tumor, they isolate the tumor infiltrating lymphocytes, they expand them and put them back, or they selectively expand them after finding the different what are some of these mutations and stimulating the T cells with those small proteins that are different and expanding them and then putting them back. And this really can work remarkably well in melanoma. It's also working quite well, for example, in lung cancer, in part because, again, lung cancer typically arises in patients who have been smokers, and smokers have, again, as smoke, unfortunately, is a mutagen. It does cause many mutations. So again, in lung cancer, there's lots of mutations, lots of differences from a normal cell, and the immune system can be trained to recognize those and go back in and actually target the lung cancer. Unfortunately, for lots of other cancers, there are far fewer mutations. And in those settings, it has become much more difficult to use this therapy where you take cells out and just expand them by stimulating with antigens that are from the tumor and generating an immune response that will then go back and work. So although that's going to be very effective in some settings where there are lots of mutations like melanoma, lung cancer, and bladder cancer, it's not going to be as successful in some other setting. Although there are individual cases, such as cases of colon cancer where there are lots of mutations, or some other diseases where individual patients have lots of mutations where this can work. In general, we're going to have to probably do something more than that to treat the majority of patients that have these kinds of malignancies. And so that's where we're going to take this conversation now, which is, what do you do if a patient really doesn't have any T cells or has so few T cells that can recognize their tumor cells that it's not really possible to expand them to large numbers to treat the patient. And so for this, our group and other groups are now doing something called T cell receptor engineering. And so, as we said, T cells have a receptor on them that allows them to recognize a target. But we and others have identified that you can identify antigens in the tumor which are potentially good targets, but the patient just may not have a receptor that recognizes it. But we can isolate by screening many donors a T cell that can recognize that foreign protein on the cell or that overexpressed protein in the tumor cell. And we can isolate the receptor from that by essentially the molecular techniques that now exist. So we can pull out the gene for that receptor, and we can now put that gene into this T cell. So we can add now a gene encoding a different receptor. So now we'll have a T cell from that patient that expresses a receptor that we know is specific for a gene that's expressed on that patient's tumor. And we know that because we've screened the patient's tumor to make sure it expresses that antigen. So we pick from this library of receptors the one that recognizes their tumor, and we put that into the patient's T cell. And now we do essentially the same process that we had talked about before, where we now expand those cells, and then we now infuse them back in. And it's important to recognize that what we're talking about here is expanding these cells outside the body and putting them back in, but making this now because we can expand them to large enough numbers outside the body. When we infuse them back in, this becomes the single most potent immune response that's present in the patient's body. So this becomes an extraordinarily potent immune response. And there's lots of antigens that we can target that are reproducibly expressed in tumors, that are expressed also in multiple types of tumors. And we can engineer cells to recognize these antigens and use it to treat patients. So our group has, for example, been looking at targeting WT1 and NY-ESO-1 and is about to start trials with MAGE and mesothelin. There are also tumors that are targeted that are induced by viruses, such as cervical cancer, a lot of head and neck cancers now, as well as anal cancer, that is induced by a virus called human papillomavirus. That is a virus where some of its genes get expressed in these cells, and that's what causes the tumor. Fortunately, those viral proteins are foreign, and although they don't naturally elicit a very potent immune response, you can isolate receptors that recognize it, and again, you can engineer cells to recognize that and use that to treat patients. And a group at the NCI has shown that they can actually target HPV-associated tumors with T cells that have been engineered to recognize HPV-associated antigens. So for T cell targeting, obviously the antigen that we're talking about must be present on the cancer cell, and we can screen for that. We can actually, by taking a biopsy of the cancer, we can do what we call profiling, transcriptional profiling. We can screen for every gene that's expressed to see whether that target protein is expressed. We know what class 1 molecules that patient presents, so we can actually screen to make sure that it recognizes. So if it's so that we can make a T cell that will be able to recognize this patient's tumor. But of course, there are some settings where we're not going to find good targets that are being processed and presented on class 1 molecules. And fortunately, to address this problem, there's an alternative strategy that's been developed, which looks at antigens that aren't necessarily processed and presented efficiently on class 1 molecules, these MHC molecules, but are themselves self-proteins that are expressed on the surface of the cancer cell. Now, T cells aren't made to recognize just proteins that are expressed in the absence of being presented in this small peptide in the context of class 1. But this is one of the things that antibodies recognize. Antibodies recognize proteins, whether they're soluble proteins or expressed on the surface of a cell. And so you can generate antibodies to these kinds of self-proteins that are recognized, or these target proteins that are expressed on the cancer cell. And if you generate an antibody to that, you can now engineer what are called chimeric antigen receptors that are composed of an antibody that actually will recognize that molecule, but will now be fused to signaling components of a T cell receptor. So it forms this chimeric receptor that has an antibody recognition structure, but a T cell signaling structure attached to it. And that is now able to recognize surface proteins in the absence of MHC molecules. And so you put that receptor into the T cell, it again still may express its own T cell receptor, but it now also expresses this. And this now is capable of recognizing targets independent of MHC. And it will now seek out and bind to that type of target cell. And as you can see, it will bind to it. And again, this receptor has been engineered to have all of the components of a T cell receptor, and in fact, some additional components. It effectively activates this killer T cell, so it again releases its cytolytic molecules, and it results in the death of the target cell. So we've now been able to broaden the repertoire of the kinds of targets that T cells, engineered T cells, can see. You may well have read about a lot of this. The therapies with CAR T cells has been approved now for treating CD19 positive leukemias. These are acute lymphocytic leukemias and chronic lymphocytic leukemias, and many B-cell lymphomas. It's also being evaluated in clinical trials for targeting CD22 or a molecule BCMA in multiple myeloma, as well as several antigens that are expressed by solid tumors. These are all in development and tested with a lot of very promising clinical results. This has already been approved, and indeed several companies have been formed that are actually are making this feasible for patients everywhere now by getting cells in, modifying them, shipping them out. So it's not just being done in a few academic centers. These are approved clinical therapies. And we're talking about therapies now that can take patients who have failed essentially all other therapies and can now respond very effectively and get complete responses. And of course, it's still early days, but we believe this is likely to result in cures for many of these patients. Many patients are now several years out after receiving such therapy. Now, we and others are now looking at trying to find ways to make these cells work better. So not surprisingly, cancers have, and we talked about this somewhat earlier on, have developed many tricks to essentially stop an immune response from working. That's one of the reasons the natural immune response doesn't work, is because cancers have lots of strategies by which it can evade an effective immune response. And we are using something called synthetic biology, where we can essentially modify genes. We can create proteins that don't naturally exist by creating genes to encode new proteins that have the capability of functioning in ways that normal proteins don't. So, for example, one of the ways that tumor cells work to turn off an immune response is they express a molecule called Fas ligand, which binds to a molecule that's normally expressed on a T cell that called Fas, and it actually results in killing that T cell. Now, there's a reason why these molecules exist. It wasn't created for tumor cells to be able to kill T cells, but this is something that exists throughout the whole body, and it's part of the way when, for example, if you injure yourself and cells start expanding to replace the injury, for example, if you cut yourself and new epithelial cells go in, it's the way they learn to stop. There are ways in which they get turned off, and you stop the cells from proliferating by using this kind of mechanism. But of course, cancer cells have co-opted this mechanism as a way of killing T cells to make them die. So what do? That may be useful. That may be useful in a normal immune response to stop T cells from proliferating when the response gets strong enough to turn it off. But it's not what you want when you're trying to fight a lot of cancer cells. And so what we have been looking at, for example, is to change this Fas molecule, to modify it. So now, as you see, the part that's inside the T cell is different. So rather than when it binds to Fas ligand delivering a death signal, we've changed that now, so that now when it binds Fas ligand, it delivers an activating signal. So this kind of immunomodulatory fusion protein, or others call it a switch receptor, now instead of killing the T cell, delivers an activation signal. So as the cancer cell starts trying to use these mechanisms as a way of evading an immune response, we actually are now turning it into a way of the immune system saying, oh, that's an activating signal. I'm supposed to get stronger, make more of myself, and actually go on and kill the cancer cell. And of course, it's not just Fas. There's one of the drugs that's become really effective in cancer therapies is PD-1 blockade, blocking these checkpoints, which again are ways in which immune responses are regulated to limit them so we don't have too much inflammation. Again, that's really important in the context of some infections. It's not very useful in the context of cancer. And so typically antibodies are given to disrupt this pathway so that the T cell doesn't get an inhibitory signal and get turned off. It gets this stop signal. But again, we can engineer this PD-1 molecule so that instead of being a stop signal, it provides an activating signal. So now, as tumors try to escape with this by expressing this PD-L1 to turn off a T cell, we actually turn that into, again, an activating signal. So the T cell interprets this escape mechanism by the tumor as actually an activating signal that says, no, keep going. This is time to be further activated, which is really what you want your T cells to do in the context of cancer. And it now goes on and kills the cancer cell. So we can engineer these cells in these multiple ways now to essentially overcome the tricks that tumors use to evade a natural immune response and to actually use those tricks against the tumor itself. So we can essentially trick the cancer rather than have the cancer trick the immune system. And of course, one of the real advantages of this is that one of the problems with checkpoint blockade is when you deliver antibodies, you're blocking this through the whole immune system. And there are some potential autoimmune consequences because some of these off signals are one of the ways we control cells from causing autoimmunity. But in the context of engineered T cells, we're putting this gene into only the cells that we've made specific for the tumor. So we get rid of this risk of autoimmunity, and we make this kind of signaling unique to just cells that can recognize the tumor. So we can bypass a lot of the toxicity. So of course, there's lots of challenges and opportunities in cellular immunotherapies. And we're just at the infancy of bringing this into the clinic, and it's only going to get better. We need to find more tumor antigens and tumors that can be safely and effectively targeted. And there are tech strategies that our lab and other labs are doing to accomplish that. We need to develop strategies to sustain T cell function in the tumor environment, which is very hostile, as we've talked about. It has ways, as I just showed you, some of the ways of turning off of T cells, but there are many other ways that it does it. It essentially uses all the nutrients. And so we have ways of changing the metabolic pathways T cells can use. We need to establish technologies to make cell therapy more broadly available to patients. And we've talked about the fact that this has become available now through commercialization of some of the advances made in academic labs initially to treat acute leukemias and B-cell lymphomas. But this is just the beginning. We are working to make these technologies broadly available for a variety of other diseases. And then it's going to be important in the context of our healthcare system to find ways of reducing the cost of producing and administering cell therapies. And I can tell you, we have our lab as well as other labs, and now some of the commercial groups are becoming increasingly effective at decreasing the time it takes to make these cells, the number of cells that are required, that will reduce the cost, make it simpler and easier to deliver this and capable of delivering to a much larger group of patients. So I'm going to stop there and turn this back over to Arthur.
Thank you very much, Dr. Greenberg, for that fascinating look into this really exciting field of this area of immunotherapy. So now we'd like to do a few questions to kind of delve down a little bit more into the specifics of it. So first, in the examples you presented, we were looking at cancer cells with just a single antigen, but obviously in reality, tumor cells express a lot of different antigens that could potentially be used as targets. So how do you go about identifying a tumor's antigens, and then how do you decide which antigens might serve as the best targets for T-cell immunotherapy?
So this is something, again, the way we looked years ago was incredibly slow and tedious and not so effective, but molecular technologies have really revolutionized the ability to do this now. So it's now feasible to take a cancer cell and look at every gene that it expresses. And you can look at the level at which it expresses those genes. And what we're looking for are genes in the cancer cell that are either unique to the tumor, such as mutations or what we call translocations, where chromosomes, one chromosome fuses with another chromosome and creates a novel protein at its fusion site. Or we're looking for genes that are overexpressed by the tumor so that it's expressed at such high levels on the tumor that we could find a receptor that'll recognize that high level of expression, but is incapable of being triggered by the low level found on normal cells. And also, we're looking for antigens that are important for why the cancer is malignant. So, for example, there are what we call passenger mutations in tumors that, for example, in melanoma, where you get UV irradiation, where just some normal cellular protein, what we call a housekeeping protein, may have a mutation in it. And you can target that, but that's probably not going to be found in all the cancer cells. But if you have a protein in the cancer cell that's important for why it's behaving like a cancer cell, it's very hard for the cancer cell to lose that. So we do this gene expression analysis in the tumor, and then we do the gene expression analysis in the normal cell from which that tumor came. And from that, we can find a group of proteins that are potential candidates. So we have a stack this big maybe. And then we look through the rest of the body and say, are there other normal cells that are expressing this protein at high levels, which if we target the tumor would be a problem? And that tends to shrink it down. But by looking essentially at thousands, hundreds of thousands of genes, we can increasingly find good candidate targets that we can now potentially make receptors for. We then use those proteins to generate receptors, isolate those receptors, and then try to target that in the tumor.
Great. So you touched on a little bit now with with putting into the genes, and I just wanted to ask a question to clarify for our audience. And with the strategies such as TCR engineering and CAR T cell therapy, where in the presentation it was kind of simplified, where they were just attached to the membrane of the T cell. In reality, it's a little more complex. Can you talk a little bit about how these T cells are reprogrammed?
Yeah, it is a little bit more complex, but it's almost equally easy now. So what really has happened, what we're doing essentially is putting a gene in and getting that gene to integrate into the cell so that the cell now, so every cell that cell now makes when it proliferates, it will express that gene because it thinks it's part of its own genome. And originally we used to have to do that. It was a very tedious process. But what we have learned, and we and others have learned, is that we can use strategies that viruses use to infect cells to actually introduce our genes. So the way viruses take over cells is they infect a cell because they have molecules on its surface that make a cell want to take it up. And then it's the genome of the virus then it gets released into the cell, and it integrates then into the cell, so that the cell now starts making more viruses. And that's how viruses propagate themselves in bodies, by essentially introducing their genes into the cell. So we've learned to take apart the viral genome, put our gene in, remove the genes of the virus, but use the surface of the virus to actually get that into the cell and now get it to integrate. And in fact, the most common virus that's used now is actually, we're using parts of the HIV virus to introduce the genes into cells, because HIV is so good at taking genes in and integrating into the genome. Now, the rest of the HIV genome's gone, so there's no risk of any HIV coming from this, but we can actually use these kinds of strategies that viruses evolved over millions of years to get into cells to introduce the genes.
I think it's very comforting to know that and to stress again that these viruses have been de-armed to eliminate all of their potentially bad properties. But speaking of side effects of these therapies, are there any side effects associated with T-cell immunotherapies? And if so, how common are they, and how are they addressed in the clinic?
So the side effects depend very much on the nature of the cell that's being infused and the nature of the antigen that's being targeted. So one of the problems with treating large tumor burdens with activated T cells is that when those cells get activated, they release inflammatory cytokines. So they release these molecules which induce more inflammation. And the problem is that's okay when it's a limited immune response, but when you have lots of tumors and lots of cells, that can cause a problem. And it's a syndrome that's called cytokine release syndrome. Patients can get quite high fevers, again, much like you'd imagine with a potent immune response to an infection. But because this can be so many cells at once seeing targets at once, You can also get your blood pressure can drop from the dilation your blood vessels get from this. And so we need to have ways of treating that. And fortunately, we've learned particularly which cytokines are important for this, for causing this. And there are drugs that can be given now that block that cytokine release syndrome, as well as now we're learning actually ways of preventing it. So there have been a couple of very recent important observations of exactly how that cytokine release syndrome is initiated, and there are drugs available to block that. So those kinds of syndromes will be less and less significant. In fact, for some of these therapies now, these cell therapies are being given now as outpatients rather than as inpatients because patients can tolerate it so well. So increasingly, we're starting to target those kinds of side effects. There are side effects called neurotoxicity, where in fact, because of some of the molecules being released, some patients have actually developed some severe side effects, including coma or seizures. That's a really rare event that's associated with these really rapid responses with CAR T cells. But again, increasingly, we are learning now how those events occur, and we can eventually we hope to soon be able to prevent that. Now, there is a whole other set of toxicities that are important to mention, which is that if the antigen that's being targeted on the cell is also expressed on a normal cell, then you have the risk of actually the toxicity to the normal tissue. So, for example, in the therapies that have been approved for acute lymphocytic leukemia, the CAR T cell therapies for targeting CD19, that molecule is also expressed on normal B cells in our body. Now, fortunately, and it does recognize those B cells and eliminate them. Fortunately, you can live without B cells, and most of these patients will require immunoglobulin infusions, essentially infusions of antibodies to replace what normal B cells will make. Or we're going to have ways of eliminating these cells eventually so that the normal B cells can return. And similarly, for some of the other targets, there are normal tissues that can be recognized. And again, that's one of the reasons for trying to find what we would call the sweet spot with receptors, with these T cell receptors, so they recognize the tumor but can't recognize these normal tissues that express low levels. But that's always the risk as we start, is will there be any toxicity to normal tissues? In general, those problems have been small, but not non-existent, and we need to be really aware of that when we initiate clinical trials. And that's why these all start out as experimental therapies, and it's only when we show can be safe that they get advanced to being approved therapies.
So you mentioned during your talk that one of the advantages of this approach, especially in the cancer types where the immune system doesn't naturally launch a strong response against it, is that you can expand the specific type of T cell that targets a tumor, and then make large numbers of them. And then once they're in the patient, that kind of forms the dominant immune response. So just how many T cells are required for these therapies, and roughly how long does that take to make all those T cells?
So that again become revolutionary in the last decade, or even last years, really. So, for example, cells which are targeting CD19, you need very few cells because those cells are capable of expanding dramatically in vivo in the patient after you've infused them, because of the nature of the receptor that's being put in and also the target that they're looking at. So for that, you actually need 1 million, 10 million cells. That can now be generated within 2 to 7 days in most settings. And so it's remarkable how quickly that can occur. Now, for some of the strategies that are being used, it depends on the strategy. That may take up to 2 weeks. For example, the T cells that we're generating to treat leukemia targeting WT1 with a T cell receptor, we are giving actually 1 billion cells. So we're giving a lot more cells for that because they don't expand as well after we infuse them. They expand some, but not as well. But even that cell dose now is taking us 2 weeks in many patients, sometimes as long as 3 weeks, depending on what kinds of cells we start with. So it is really becoming revolutionized how quickly these cells can be generated. And there's a real focus on making this quicker and more efficient and making these cells better able to expand after you infuse them, which means you can infuse fewer cells. So I think unquestionably within the next few years, these therapies will not take more than a week to develop and to be able to administer to patients.
Wow. I think one of the really interesting things about these is that unlike almost every other treatment, these are actually living, rather than just being an inanimate drug or molecule, these are actually living therapies. So once these T cells are reinfused into patients, how long do they last in the body, and how long can they provide protection for?
So again, that very much depends on both the nature of the cell that you've modified, as well as the nature of the target that you're going after. Because some of these tumors, particularly the solid tumors, have enough mechanisms for slowing down growth that actually eliminating cells, that we need to engineer ways to make these cells persist. But I will tell you, for example, in our acute leukemia trial where we're targeting WT1, in many of these patients, these cells are around 3 years. We now have patients 3 years out with a large fraction. Still, the most potent immune response in their body is the cell that we've infused. And that's true in a fraction of the patients with CAR T cells as well, that they can really persist quite long. A lot of it is dependent on how you engineer that and what kind of cell you put it into. And we've learned that memory cells, that if you take out cells that have been programmed in our body to become memory cells. So for example, we know when you get infected with influenza virus, you generate a lot of what we call effector cells, which kill the virus, but you also generate these memory cells, which last forever or for at least decades, and actually will recognize that virus if you get infected again. And what we've learned is how to isolate those memory cells and put our genes into those cells so that these are self-renewing, and that's the kind of cell that can last for years. And so one of the real advantages with these cell therapies is, just as you've alluded to, these are living therapies. These don't just kill and mediate their effector function, but they can self-renew and expand. And then when they stop getting stimulated, can form long-term memory and provide patients with presumably lifelong protection from recurrence.
Wow, that's amazing. So I noticed that when throughout your presentation, you mentioned that there were T cell immunotherapies, specifically the CAR T cell therapies, that were currently approved for lymphoma and leukemia patients. As far as patients with other types of cancers, what type of clinical trial opportunities are available for them? And then just if you could follow up that with talking a little bit about the importance of clinical trials for advancing these cellular immunotherapies.
Yes, I think I'll start with the last part first, which is really, it is critical that these studies obviously be done in organized clinical trials, because there are many questions to be asked and that need to be answered for this to move forward. And we can only do that really with organized clinical trials, where you can ask and answer a question, understand what the limitations are, and build on those limitations, build on that to actually overcome the obstacles to effective therapy. We are preparing, for example, within hopefully the next 6 to 8 months, to initiate a clinical trial in pancreatic cancer. We have an ongoing trial in lung cancer that we hope to expand and to develop an additional therapy now that will target lung cancer. And shortly after the pancreatic cancer trial is initiated, we hope to initiate a trial in ovarian cancer. There are trials that have gone on from other groups at the NCI that are developing therapies similarly for some other solid tumors, including colorectal cancer. But all of these are trials in development. And the only way that these can actually be developed is by essentially patients enrolling in the trials that can be done. And of course, we recognize some of this comes with some risks. And so many of these patients we recognize, have been quite selfless in essentially allowing themselves to be put on clinical trials where really questions are being asked. But I can assure you that all of these trials have been designed in ways in which we have tried to minimize toxicities. Now, we can't predict all risks and benefits, but things are not being put into patients for which there is not a really good rationale and expectation that they will provide benefit, although unfortunately we never guarantee that without actually doing it. But increasingly, the numbers of diseases that will be approached with clinical trials is increasing astronomically. And there are trials, for example, that are being initiated here with CAR T cells in patients with breast cancer, as well as there are trials that are being developed for example, at University of Pennsylvania in Carl June's group, in pancreatic cancer as well as in ovarian cancer. So we're seeing this enormous expansion of diseases that can be treated. And that's why I say I think we are just at the infancy now of these strategies working. We understand some of the limitations, and we need to move stepwise and rapidly to make these effective therapies for other types of tumors.
It's good to hear about that progress. So we've got time for one last question. And with that time, I was hoping that you could give us your outlook for cellular immunotherapies moving forward and how you think that they will impact the future of cancer treatment.
Clearly, in my mind, the real benefit of cellular immunotherapies is going to come as we get more and more creative with the use of synthetic biology. So we can not only find targets, we can find increasing targets to go after, but we need ways of making these cells capable of maintaining function and working against solid tumors. And we are increasingly understanding how solid tumors evade immune responses, and we can build in genetic pathways into these cells. We talked about some of them, like the modified Fas molecules and PD-1 molecules, so that these T cells will be able to take advantage of the way tumors try to escape an immune response and use that to actually drive their own activation and maintain their function in these tumor environments. We can also make T cells capable of being able to use nutrients that are available in the tumor microenvironment that normally they can't use, that tumors are using to stay alive, but that T cells don't use very efficiently. And we can build in these metabolic pathways into T cells, so they will use them, and they can then compete successfully with the tumor for these nutrients and use them to actually fuel their own growth, and in growing, use them actually to go after and kill the tumor cell. So the advent of synthetic biology is not only going to make it possible for T cells to work better, but it's also going to be able to make them be safer. So we can, for example, build in what we call on-off switches into T cells, so that if they are causing a problem, we can turn the receptor off so they stop functioning for a while, and then turn it back on when they're necessary. Or we can have what we call a suicide signal, so we can eliminate cells if they're toxic, so we can guarantee that they will be safe. And we can have what are being called, developed initially by Wendell Lim at University of California, San Francisco, these AND signals or signals, where a T cell has to see two molecules on the tumor to be effectively triggered rather than one. And if we do that, we can really make these T cells capable of only seeing the cancer, because it takes advantage of proteins that are two proteins now that are expressed, so that it can't really see a normal cell which only expresses one of them. So you can take advantage of essentially expanding the breadth by which a T cell, what's required for a T cell to see the tumor, but make it very selective for the tumor. And so there are all these ways of building in genetic signals into T cells that will make them increasingly specific. So cellular immunotherapies are going to become, in some ways, a bit more complex because of the genetic programming, but because we can introduce all these cells typically in a single vector, it won't change the time to generate the cells. It won't make it much more complex. It just makes the off-the-shelf reagent will have to actually be made in advance. And so I think the use of cellular therapies is really at its infancy, and it's something that I think has an enormously promising future.
Excellent. Well, that is all the time we have for today. Thank you so much, Dr. Greenberg, for your extremely informative webinar. It's great to hear about the progress that's been made in this exciting area of cellular immunotherapies. I know I learned a lot, and I hope that those watching out there learned a lot as well, too. So for more of our webinars and the additional resources that the Cancer Research Institute has for patients and caregivers as part of our Answer to Cancer educational programs, we encourage you to check out our website at www.cancerresearch.org. Here you can read and watch stories shared by others who have received immunotherapy treatments across a wide variety of cancer types. You can register for one of our immunotherapy patient summits. You can browse our entire library of past webinars featuring the world's leading immunotherapy experts, such as Dr. Greenberg. You can access information on other resources, including treatment, emotional support, and financial assistance. And you can find help in locating an immunotherapy clinical trial. On June 30th, we kick off our Immunotherapy Patient Summit series in San Francisco. At these free half-day events, we connect patients and caregivers with immunotherapy experts to discuss the latest advances in research and treatment. Other summits are scheduled for New York City, San Diego, and Houston. Please go to cancerresearch.org/summits to learn more and to register to attend. Finally, I'd like to thank our sponsors one last time for making this webinar series possible: Bristol Myers Squibb, Regeneron, Sanofi Genzyme, and Adaptimmune. I'd also like to recognize and thank BioRender, whose platform was used to create the majority of the graphics that you saw today. And again, you can watch this and all of our other webinars on our website at cancerresearch.org/webinars to learn more about the immunotherapy options that are available in a number of cancer types. Dr. Greenberg, I just want to thank you so much again for taking the time with us today and for the amazing work that you are doing to help patients. We wish you the best of luck.
Thank you.
Awesome. Well,